Okay, so I may talk a bit fast. I've got a lot of info here. The title of my thesis is Temporal Microplastic Concentration Fluctuations within an Agricultural Creek and Groundwater in Relation to Farming Practices. So we know that there are microplastics in agriculture. Some of the more common places we find them are seed coatings, organic fertilizers, agrochemicals, tile drainage, silage bags, things like that. A little background on the agriculture in Illinois. We have over 71,000 farms that cover 26.3 million acres. We're the largest producer of soybeans and the second largest producer of corn in the US. Those two crops alone cover 21.6 million acres in Illinois. The majority of these crops are treated with pesticides and fertilizers each year, and about half of each of them have manure applied. We do have some use of irrigation, and about 8.8 million acres have tile drainage. For the purpose of my study, I split the year into farming seasons, and these are the seasons and the practices. Winter has basically no activity. The planting season has tilling, fertilizing, planting, and application of agrochemicals. The growth period has irrigation and second and third applications of agrochemicals. And then the harvest is obviously harvesting of crops. And then we have tilling and fertilizing in preparation for spring planting and also the planting of cover and winter crops. Now, this is strictly for corn and soybeans in Illinois. This can vary with different climates and different crop types. So my research questions, do certain farming practices add more microplastics to the environment? And are there farming seasons that contribute higher concentrations than others? My research area was Kilbuck Creek, which is located in northern Illinois. It is a 53-kilometer long creek. It flows south to north primarily through agricultural land. It does intersect three counties, DeKalb, Ogle, and Winnebago, and ends at the confluence with the Kishwaukee River. For my site selection, I chose 5 sites along the creek. My sites are these red triangles. I have one tributary site and one groundwater well site. These yellow shapes are landfills. My sites were selected due to accessibility and location along the entire creek. I pumped about 20 liters of water from each site using a peristaltic pump. I connected repurposed motorcycle fuel filters that had different size mesh screens in them, attached with flexible tubing. So I pumped the water and the microplastics were then collected in these filter housings right here as I collected. So then I only had the hose and filters to take with me. After that, I digested organic material, put the samples through density separation, filtered them, and counted them. My hypothesis was that microplastic concentrations would increase downstream and during the planting and growth seasons due to increased farming activities. What I found was that overall microplastic concentrations decreased downstream. So here on the y-axis, we have the sites. On the x-axis, we have the sampling months. We started high in March at 18.6 microplastics per liter. We had a dramatic dip in May, and then we went back up in July and steadily decreased throughout the year. As far as going downstream, we started very high at 21.9, and it steadily decreased, except at 5C, where it increased again. And this has happened in every sampling round. The tributary was well below average, as well as the well. The exception here is in September for the tributary and March here for the well. So some potential reasons why we see this decrease downstream. Most other studies have seen an increase downstream, but they attribute that to the proximity to urbanization. This is a land use map of the Kilbuck Creek area. The yellow is corn, the green is soybeans, and this gray is urbanized areas. We have a severe lack of urbanization, which is great for my research. The closest proximity of urbanization we have is here. This is Lindenwood, which is a town of about 600 people, so not a very sprawling metropolis. Um, Another potential reason I see a decrease is I may not have had enough sampling sites. And also we have quite a large distance between some of these sites. So there may be something that had happened there that is not in my results. And then we do see this increase here at 5C. This is after the landfills, which are located here. So if the landfills weren't there, we may also see a decrease down here, but this potentially affects the concentrations. And then we have sinuosity. So as you can see here, this is just a very small portion of the creek and it meanders very much. With that meandering, we have an increase in point bars. Point bars is the area where we get deposition from the creek. So an increased amount of point bars potentially means that we have microplastics being deposited on the banks and therefore not making it downstream. Physical characteristics, there's a study by Drummond et al. 2022 that says that narrow, shallow, and slow-moving bodies of water have a higher or a longer residence time for microplastics within them. Kilbuck Creek is extremely narrow, very shallow, and slow-moving. So this could also potentially delay that transport downstream. And then Kilbuck is highly vegetated, both along the banks and within the creek. So vegetation acts as a filter or a trap for those microplastics. Shape and density trends. Here we see fibers account for the majority at 72.5% of all microplastics collected. Fragments came in second at 24.7%, and we see that ratio at every single site. The only exception we see with foam, film, and pellets, which are pretty much negligible, is here at 1C. We have higher than average amounts of pellets at this one location. So something is happening there. That tells me that there is some outside influence that is happening. Upon further investigation, there was a construction supply company located about 1 kilometer upstream from that site. So there potentially is something there that is affecting the shape distribution at 1C. Here we see the density trends. The most abundant densities were the largest density and the lightest density. The middle were lower. So this tells me that the same materials are being used season over season. We can see this consistent kind of soup bowl shape. There's a couple outliers, but for the most part, we see that same trend. And then precipitation. So 2023 was a drought year. The blue line here is 2023. The red line is a 10-year average of precipitation. And for most of the year, we were well below average. We have a couple that are above average. The one we want to look at is July here. So the bottom, we have our microplastic counts. In the top, this is a weekly precipitation average. Here we see a dip in May, and we also see a lack of precipitation here. And we see this spike in July, as well as this spike in precipitation at the same time. There's also a spike here in October, but I did not sample, so that is not reflected in my data. So this got me thinking that precipitation is potentially the driving factor for the microplastic fluctuations. So what I did was I added the cumulative total of rainfall 2 weeks before sampling, 1 week before sampling, and 3 days before sampling, and compared those with the microplastic counts. As we can see here, these are just 2 of my sites, but there is no correlation between precipitation and microplastic amounts. So precipitation is not the driving factor here. The tributary and the well have different dynamics. The tributary is channelized, likely has tile drainage. We see that the spike here is in September instead of July. This could fall back to that Drummond study. The tributary is narrower, slower. There may be a delay. And then the well is not located on farmland, but it is adjacent. It's subjected more to surface runoff and atmospheric fallout. We can see that reflected here in the fibers. Fibers are more easily transported by the atmosphere and surface runoff, and so we see a higher amount of fibers within the well. There are several limitations. The scope of this project was not realized until I got into it. I'm unaware of any previous studies that have related microplastics to specific farming practices or seasons. So this makes it difficult to have any kind of comparison or confirmation of my results. Secondly, there's no standardized methods for microplastic research. So again, difficult to compare. My analysis was visual. There are more advanced methods of classification, but those were not available to me. Sampling sites, I was limited to accessibility. I may not have a complete profile of the creek. Sediment samples were not taken, and this could potentially establish the potential for settling or deposition of those microplastics. Again, we had an anomalous year with the drought, so an average precipitation year may show different results. And then farming practices can vary, as well as the products and the equipment used. Future research would benefit from analysis of several fertilizers, seeds, equipment, and then also a survey of surrounding farms on what their practices are and when they take place would be very helpful. And again, my focus was on corn and soybeans, so other crops have different agricultural needs. So again, future research would benefit from a long-term, geographically diverse and crop-inclusive study. So in conclusion, the overall trends in my concentrations between the sites over the year indicate farming activities could influence the concentration of microplastics. The consistencies in shapes and densities indicate the same products and equipment are being used year over year, season over season, and having no correlation to indicate precipitation is the driving factor. Ultimately, I cannot determine with certainty that farming activities or seasons are solely responsible for the fluctuations, but are definitely a contributing factor. And that's that.
Great. Thank you, Jennifer. We do have a number of questions, so
Okay.
Yes, I
I'll
can
turn it over
get
to
us
you.
started with the first one. Are there differences in the irrigation strategies for the different crops that may influence runoff to the water body?
That's speaking to something I'm not entirely sure of. There's not a lot of irrigation in my area, so I'm not extremely familiar with the practice. So unfortunately, I'm not able to answer that properly.
Okay, and this one is the sort of related. It says, adding to the question before this one, are there differences in what is being applied to the fields, not only irrigation, but amounts and types of things being applied?
Yes, and a lot of the information I found was from the agricultural census from the USDA. Different farmers have such different practices. Some apply only organic fertilizers, others apply agrochemicals. Different crops have different needs. Corn is a very water intensive crop. So there's more water applied when you have irrigation. Other having cover crops can eliminate some of the fertilizing applications. So it varies so much. And again, as I said, the scope of this was not realized until I got into it. So there's so many factors with just farming practices that would need to be analyzed and applied.
Great. And I think this one is also related to that in how do you know that these plastics are attributed to agriculture? Are there certain plastic types they use in the industry?
So there are, again, that would require a lot more analysis. Definitely a material scientist would be useful to analyze different fertilizers, pesticides. We see the microbeads in fertilizers that have the time release formula. So that plastic kind of breaks down and that's what releases those chemicals. But yeah, I'm so sorry. My brain just kind of, what was the exact question again?
Yeah, so it was how would you know that these plastics are specifically attributed to agriculture? Are there certain plastic types that they use?
Okay, yeah. So finding out the plastic types would be a material scientist that would be helpful. As far as the Yeah, that's where I would probably end up saying that we would need more analysis. For my research specifically, because there was the most likely culprit is agriculture. Future research is going to be, it's going to be difficult because urbanization does affect so many waterways. But again, that's a lot of future research.
Okay, and we do have two more questions. Sarah, do we have time for those?
Yeah. We're right at time. I think since we're going into lunch, I think we can take these two questions, and if folks have to drop off, they can go ahead and drop off for lunch. But keep in mind that if you're dropping off, that the speed networking starts at 12 o'clock if you'd like to join, but we'll be back at 1 o'clock for lunch. But yeah, go ahead and let's do those two questions.
Okay, great. So the first question is, what was the method size detection limit? You reported your results in concentration. However, the units of number over liter does not truly reflect concentrations. Finding with lower counts could actually be higher in terms of mass in cases where the pieces of plastic are large. Were there detailed size measurements of the microplastics performed?
Sure. So I had 3 different mesh sizes, 400, 250, and 125 microns. For my study and my results, I used the 125 micron mesh screen. So that's extremely small. Also, my filters, I think, were 0.45 microns. So we're looking at very, very small microplastics. The larger ones were not included in that particular result. There's definitely a factor of missing some things, especially when you have the deposition in the sediments and things like that. So I was somewhat limited to just those sizes, but I'm sure that there are even smaller microplastics that I was not able to get.
Okay, great. And the last question here is, do you think that the microplastics derived from biosolids could be a large source for your study?
It definitely. We know that the wastewater treatment plants typically will give those biosolids to farmers for a very small cost or even free. And that just goes right back into the agricultural soils, which then we know that microplastics are crossing the cell wall barrier and getting into our foods. So it just becomes a cyclical cycle of microplastics going from biosolids back into our foods and back into us. Also, livestock feed has plastic fillers in it. So the organic fertilizers from livestock is definitely additive to that.
Great. Thank you so much for answering all those questions and
Yeah, thank you.
staying on. I appreciate it.